Hmm?
Okay, dear student, in last class, I have discussed about the the second type of receptor, that was ligand ion channel receptor. That was our third topic, and today we will discuss with the fourth classification of receptor, that was Jak-Stat receptor. As I told you in transduction mechanism, receptor are five types: G protein coupled receptor, enzyme linked, ligand ion channel, Jak-Stat, and other receptor that generally helps in gene expression. I have told you in G protein coupled receptor and gene receptor controlling gene expression, these two are the main receptor which we have to focus more, but not that you will not focus in other three, that other three are also important. In that, one of the most important is Jak-Stat. The Jak-Stat is similar to kind of what you have studied in enzyme linked in receptor. That receptor work and Jak-Stat receptor work is almost same. Some difference are there that I will tell you in that video, and I will tell you how they differ and what is their similarity and why they are different, and what are the proteins present there and here, and what is the difference between that protein and this protein, and how Jak-Stat receptor is going to work and show its action by binding with different drug in our body. So let's get started. So in that, I have drawn a diagram of Jak-Stat by which, so that you can easily understand Jak-Stat, like remaining and other receptor like ion channel ligand receptor and enzyme link receptor. Jak-Stat receptor are also present inside the membrane, just similar like ion channel and the enzyme link receptor. But what is the difference between enzyme link receptor and Jak-Stat receptor? Here, there we saw two domains are there, but here generally external domains are there, but in internal no intrinsic catalytic pathway or domain is there. That's why Jak-Stat is different than ion channel receptor. In ion channel receptor, they have intrinsic catalytic domain. That's why that also is calling as catalytic receptor. So other name of ion channel receptor or enzyme link receptor is catalytic receptor. But here in Jak-Stat there is no domain, such domain where intrinsic catalytic pathway takes place. So there is no catalytic internal domain or intrinsic domain. That's why Jak-Stat are different than ion channel and enzyme link receptor. Let's go into the deep of Jak-Stat, where we will study how that Jak-Stat is going to work, what are their example, and how it looks like by easy simple diagram. So I have drawn here, that is a Jak-Stat receptor, that is a one receptor. Like that, enzyme link receptor, we saw that inside there was a enzyme link domain. Here also enzyme link or enzyme binding domain is there, and where there we saw that main enzyme that is going to be bind with that receptor, that is tyrosine. Here also the same thing in Jak-Stat receptor also. With that receptor, the most common enzyme that is going to link in enzyme binding receptor or enzyme binding domain, that is tyrosine. So here also tyrosine is added or binded with Jak-Stat receptor when it is in steady form or free form, or they are not working. But whenever cytokines is going to come and going to join into the binding site of the receptor, then that Jak-Stat receptor will get activated and they started to show its action. So when cytokines are not added, they are in free form and steady form, they are generally inactive and they are binding with a enzyme, especially tyrosine. But when cytokines is coming and bind into the active site of the receptor, they are getting excited and they started to show its action. So what it will happen in that activated form, JAK kinase, what is also known as genus kinase, that is a tyrosine dependent kinase that we have already studied in enzyme linked receptor also. There we studied RTK, same like there we studied RTK, but instead of RTK in Jak-Stat receptor, that kinase is called as genus kinase, or in short that is denoted by Jak. But last time we told us that is receptor tyrosine kinase, that's why that name was RTK, but here that is called as genus kinase, also stated as Jak. So in ion channel receptor, we showed that whenever that receptor is getting excited or activated, one protein will come and bind with Jak, or bind with the kinase that is present. There RTK kinase was present, so with RTK different protein came and binded, but here in Jak-Stat, instead of RTK, Jak protein is there. So to bind with Jak, one different type of protein will come. That protein is called as STAT. What is the full form of STAT? STAT means signal transduction and activation of transcription. So that protein will come and will bind with Jak and will form a complex. That same thing will happen in that receptor. Like enzyme link receptor, I told you, two receptor present nearby, they will work each other, help each other, and they will help the total receptor to show its action. Same like here also, Jak-Stat, two receptor will help each other and help each other to show all over action of the receptor. So now I will discuss after binding with Jak and Stat what is going to happen. I have told you two receptor is present nearby. Suppose that is one receptor Jak-Stat and another receptor is there. In that receptor also one kinase is present, that is genus kinase. In ion channel I told you that was RTK, but in case of Jak-Stat that is called as genus kinase or Jak. So here I told you there also different protein came and binded with Jak, binded with RTK. Here also one specific protein will come, that specific protein will come, that is Stat. I told you what is the meaning of Stat. Stat means signal transduction and activation of transcription. That protein will come and will bind with Jak. Here also same thing will happen. Near Jak, that Stat protein will come and bind with Jak. After when they are binding with that Stat, they are going to be phosphorylated. When they are going to be phosphorylated, that phosphate will come and will bind with Stat protein, and here also that same thing will happen with Stat and Jak complex. Phosphate will come and it will cause phosphorylation and it will bind with Stat. When that Jak and Stat is going to bind with protein, bind with phosphate, they both get activated. After getting activated, that both Stat with phosphate will separated out with Jak, and after when they will separated out with Jak, that two Stat what is remaining, that two Stat will bind with each other. And you have to remember, when the Stat is going to be removed out from Jak, they will only be removed with Jak, but that phosphate will remain with Stat. So when they are removing from Jak, that two Stat with bind with each other with phosphate, in phosphate activated, connect and binded with it, the Stat will connect with each other and form a dimer. When they forming a dimer, that is leads to the production or activation of phosphate ion or phosphate or phosphorus. So what it will happen, that will cause the activation of Stat protein. When that Stat protein will get activated, that Jak-Stat receptor will get activated, and then they will help in showing their action. Their main action is transcription. So when they are going to be activated, they are going to show its action, they are going to be activated, and they are going to show its action, which is main transcription, and that is leads to their main action, that is transduction mechanism. So by that Jak-Stat is going to work, that I already have told you that is have many similarities with ion channel and enzyme link, that I've already discussed when I was describing that mechanism of action. So now I think it is clear for you. Now I will tell you what are the examples of Jak-Stat. What is the example of Jak-Stat receptor? We know interleukins and cytokinins, that both receptor comes under Jak-Stat receptor. So what is Jak-Stat? Jak-Stat also one receptor which is present in membrane. When they are inactive in form, they are binded to a enzyme in enzyme binding domain, that is tyrosine. In that case, any other catalytic intrinsic domain will not be there, only enzyme binding domain will be there, where the tyrosine will come and bind in inactive form. But when they are binded with cytokines, means when cytokine will come and will bind with a receptor, that receptor will get activated, and it lead to the activation of a special kinase protein, that is called as Janus kinase, which is a tyrosine dependent protein kinase, also called as Janus kinase or JAK, that is going to activate it. When the JAK is going to be activated present in two nearby receptors, they are going to activate it, and they are going to attract a stat molecule or stat protein, which is called as the signal transduction and activation of transcription. So that protein will come, and that protein will binded with JAK. When the JAK and stat forming a complex, they are going to be activated, and then phosphorylation takes place, and phosphate will come and will bind with Jak-Stat combination of Jak-Stat complex. When the Jak-Stat and phosphate is making a complex, they are going to be activated, and what it will do, it will remove out Jak and stat, and it will be get separated. When the Jak and stat will get separated, the stat already binded with phosphate will come together and will bind with each other and form a dimer. When they are getting binded and forming a stat dimer, they are going to be activated, and it leads to the activation of Jak-Stat receptor, and it leads to their main work, which is transcription, and it leads to their main mechanism, that is transduction mechanism. And I also told you some example of Jak-Stat, in that interleukins 2 and cytokinins receptors are there, which are comes under Jak-Stat receptor. So by that I have easily explained you what is Jak-Stat, what is the main structure of Jak-Stat, how it is going to act, what is the mechanism of action, how it is same or different than ion channel receptor and enzyme link receptor. Like that I have described what is Jak-Stat receptor. In upcoming class I will be discussing with the last topic of receptor, that is receptor helping in gene expression, into the upcoming videos. To get my notes for Jak-Stat receptor, I have already prepared notes. That notes link is already given into the description. Go and check it out, and check what is Jak-Stat, how it is working, and tell me what is their problem or not. Go and check it out from the note that you can download from the description. For today it's okay. Meet into the next video. Thank you.